Good morning. My name is Peter Cannon. I've been with the HPRA since April 2021. And prior to that, since 1986, I've worked in industry in a number of multinational companies. And I was a member of the leadership teams of these companies with responsibility for process development, technical support, and QC functions. So this morning we're going to discuss out-of-spec investigations. Initially, we're going to have a look at the regulatory background, some potential consequences, the what ifs if we don't manage our out-of-spec investigations appropriately. We'll have a look at the work processes involved. We then take a look at examples of some deficiencies, and then we'll finish off with some takeaways. Okay, initially looking at the regulatory background. In Europe, obviously, UGLEX Volume 4, Part 1, the basic requirements for medicinal products. Chapter 6, under quality control, in the section on ongoing stability programs. Section 6.2 speaks of out of specification or significant atypical trends should be investigated, and any confirmed out of specification results or significant negative trends should be reported to the relevant competent authorities. The possible impact on batches on the market should be considered in accordance with Chapter 8 of the GMP Guide and in consultation with the relevant competent authorities. Annex 16 in Section 1, where it deals with certification, Section 1.716, all investigations pertaining to the batch being certified, including out of specification and out of trend investigations, have been completed to a sufficient level to support certification. Now, moving on to UGLEX Volume 4, Part 2, which deals with active substances used as starting material for drug products. In Section 6.6, under laboratory control records, 6.61, complete records should also be maintained for out of specification investigations. 6.7, batch production record review. All deviation investigation and out of spec reports should be reviewed as part of the batch record review before the batch is released. In Section 8.3, dealing with in-process sampling and controls, 8.36, out of specification investigations are not normally needed for in-process tests that are performed for the purpose of monitoring and or adjusting the process. There is one exception to this, and we'll deal with that later on in the presentation. In Section 11.1, under general controls, 11.15, any out of specification results obtained should be investigated and documented according to a procedure. This procedure should require analysis of the data, assessment of whether a significant problem exists, allocation of the tasks for corrective actions and conclusions, and any resampling and or retesting after out-of-spec results should be performed according to a documented procedure. And to my mind, if you stick with the principles in that single paragraph, I don't think you're going to go far wrong when you're dealing with your out-of-specification investigations. So let's move on to some potential consequences to mishandled out-of-spec investigations. There can obviously be serious potential consequences to poorly managed out-of-specs. One thing we're all very concerned that we don't have any impact is that there is any patient harm. We can have an impact on stability programs. It can result in quality defect reports, customer complaints, the potential of product recalls, and this obviously can have a major impact on schedule. It results in increased surveillance with more frequent regulatory audits. And I think the thing to remember is that in all of this, the lab is pivotal to early investigation of issues. Let's move on to the work processes itself. Why do we need to investigate out of spec results? We need to investigate out of specs to determine the root cause. Is it lab or manufacturing related? The investigation itself should be thorough, timely, it should be scientifically sound and well documented. It's important that we don't discard test samples, and we carry out of spec investigations for batch release testing and of starting materials. And here's the exception we mentioned: in process control testing, if the data is used for batch calculations, decisions, or if included in dossier or on a certificate of analysis. For stability studies on marketed batches of drug products or APIs, and in cases where previously released batches used as reference samples in an out-of-spec investigation themselves show out-of-spec or suspect results, and for batches undergoing clinical trials. Okay, so let's move on to initially phase 1 investigation. Initially in phase 1, we're looking for obvious errors due to external consequences or to the analyst has noted prior to generating data. Again, I just want to re-emphasize that it's very important that we don't discard samples before batches are approved. If no error is found in this preliminary phase of the investigation, we need to expand the investigation. And just before we moved on to the expansion of the investigation, I'd just like to measure investigational measurements. The use of investigational measurements is at the discretion of the QC supervisor and is focused on what, if any, lab error could have caused the out of spec or out of trend if we suspect that it is, in fact, a lab-based failure. It's very important to remember that investigational testing may not be used to replace the original suspect analytical results. It may only be used to confirm or discount a probable cause. So, moving on, in the initial investigation is conducted by the analyst and the supervisor, and often makes use of lab investigation checklist. It is important that we may involve interested stakeholders, example, production, QA, contract giver in the case of CDMPs, or the MAH or QP. The analyst and the supervisor, the investigation, it should be restricted to the data, the equipment, and the analysts, the review of the analysis itself only. On completion of the documentation review, remeasurement can occur once the predefined test plan is documented, and it's very important to remember that this is only to support hypothesis testing. This initial hypothesis testing can include the original working stock solutions, but should not include preparation from the another preparation, excuse me, from the original sample, i.e. retesting. So it's typically refiling, redilutions, and reinjection of the individual sample at this early stage of the investigation. Moving on to phase 2, if we haven't established a probable root cause or any assignable cause in phase 1, we will move on to the phase 2, which is a more expanded investigation. It's important at the outset of the phase 2 investigation that we, if we haven't already, we contacted the other stakeholders, and that primarily here it's carrying out a manufacturing review. We've already mentioned hypothesis testing. That can be applicable to phase 1, but it is also applicable in the phase 2 investigation. But prior to any additional phase 2 hypothesis testing, it's important that we carry out the manufacturing review. When we do carry out the additional hypothesis testing in this phase of the investigation, again, it's important we document everything. The description of the testing it needs to be documented and approved by QA, possibly the contract giver or a QA equivalent, prior to initiating the investigational testing. The requirements of the investigational testing: the description must fully document the hypothesis to the test, the theoretical root cause being investigated, what samples will be tested, the exact execution of the testing, how the data will be evaluated, and this hypothesis testing, it may continue from the remeasurement of the original preparations in phase 2. But again, it's important to remember that this hypothesis testing can't replace original suspect analytical results. It may only be used to confirm or discount probable cause. If no assignable cause has been elucidated that could explain the results during the manufacturing investigation or the assay failure investigation, retesting may be considered. Part of the investigation may involve retesting a portion of the original sample, not a different sample. This is important to remember. The decision to retest should be based on sound scientific judgment. Again, we need to generate a test plan. It needs to define the number of retests, and this number must be statistically valid and performed by a different analyst. It's important to remember that all retest results must meet specification. We can't rely on averages, and we resample only in very exceptional circumstances. If the batch is rejected, if we confirm the out-of-spec result, we still need to complete the investigation to see if other batches are impacted and to identify and implement CAPAs. But the decision to reject, is important to remember that the decision to reject, it cannot be reversed as a result of further testing. The impact of the out-of-spec result on other batches, ongoing stability programs, validated processes, test methods, it needs to be considered and documented in the conclusion. We mentioned resampling, and we have had a number of queries about resampling. So when we resample for retesting, it should only be, as I've said, under exceptional circumstances. The original sample should be sufficiently large to permit additional testing in the event of an out-of-spec. If investigation concludes that the original sample was not representative of the batch or was compromised in some way, if resampling is justified scientifically, it should be performed by the same qualified method that was used for the initial sample. If the investigation concludes that the initial sampling method was deficient, a new sampling method should be developed, qualified, documented, and approved by quality management. It's important to remember that resampling, when justified, always must be approved by quality management. So now we're going to move on to some recent deficiencies. We'll start with deficiencies relating to the out-of-spec procedure itself. Initially around hypothesis testing. The hypothesis testing didn't require formulation of hypothesis for retesting, where no obvious lab assignable cause identified during the initial lab investigation. The procedure did not explicitly state quality management approval was required prior to proceeding to hypothesis testing. The procedure did not clearly state that hypothesis testing was used to identify a potential lab root cause, and such test results could not be reported as the final result. Looking at manufacturing investigations, did not clearly document at which stage a manufacturing investigation was initiated, and there was no assurance that such an investigation was completed prior to formal retesting. Again, sticking with deficiencies relating to the out-of-spec procedure, retesting. The procedure didn't specifically state that retesting was required to be documented and pre-approved by quality management. It did not require documentation of rationale for formal retesting being carried out by a second analyst. They allowed for averaging of retest results, but did not explicitly state that all such results must be within specification. Deficiencies relating to the out-of-spec procedure. Reanalysis retesting was performed in lieu of investigative testing, yet these results were averaged and reported in a number of cases reviewed. Resampling did not require prior documented approval by quality management. The procedure did not specify a timeline for the completion of the laboratory investigation. The date of discovery was not recorded on the out-of-spec form, and therefore it could not be verified that the record was raised in a timely manner. Looking at some example deficiencies themselves, hypothesis testing not documented and justified. The investigation was deficient as follows. Retesting as part of the secondary investigation included testing by a second analyst, repreparation of standards due to suspected glassware contamination, change of mobile phase due to suspected contamination, change of column due to suspected column contamination, and change of the HPLC instrument. However, there was no documented evidence within the report that any of this retesting had been pre-approved. Again, looking at an example of a deficiency around hypothesis testing, not supported by data and justified. With respect to an out-of-spec for individual impurity and total impurities, the following was noted. The performance of retesting as part of the phase 1 investigation was not suitable or in line with the company's procedure, as an obvious root cause had not been determined. The laboratory investigation concluded that the out-of-spec was due to dirty glassware. However, there was no evidence to support this conclusion, and no CAPA had been raised to address this issue. An investigation relating to a low assay was deficient as follows. The original out-of-spec results were invalidated excuse me, based on duplicate retest results only, and the hypothesis that the HPLC column was the root cause was not supported by evidence, in that no hypothesis testing had been performed to demonstrate that the column was the source of the issue. The data from the reference batches tested as part of the investigation conflicted with the hypothesis that the column was the root cause for the low assay result. Looking at a deficiency example around investigative test results being reported. In a number of cases where in-specification results were obtained during hypothesis testing, these results were used as a reportable result. For example, regarding the investigation relating to an out-of-spec for assay, the hypothesis that the root cause of the out-of-spec result was an instrument issue was not proven. The hypothesis that the repeat analysis result was due to addition of excess material during weighing was not tested and proven as part of the investigation. An in-specification result from the second repeat analysis under a different hypothesis, out-of-spec result was due to a problem during extraction, was used as the reportable result. Looking at an example where there was a lack of robust impact assessment. The investigation of regarding product A, batch 1, which was out of spec for low assay, was deficient in that the investigation indicated that the out of spec result was valid and no root cause could be identified. However, no consideration was given to the potential impact on previous lots of product A released. No reassessment of the risk associated with a previous investigation relating to an out-of-spec for the same parameter for a batch of product A was performed in light of this out-of-spec. And it's important to note that the batches in question here have been manufactured consecutively. An example of a deficiency where resampling was neither documented or justified. The investigation relating to a higher than normal level of residual solvent in the product, including resampling and retesting of individual drum samples of the product without a documented rationale for resampling having been approved by quality management. The investigation relating to an out-of-spec moisture content on the composite sample of material D, including sampling of all individual drums of the batch. The individual drum samples were retested and 2 drums with high Carl Fisher were removed from the batch. The batch was released based on testing of the composite of the remaining X-2 drums. The resampling and retesting strategy had not been documented in a protocol approved by quality management, and there was no documented justification for the deviation from the standard sampling and release process. Now let's just see what takeaways we can take from the issues we've been considering. An out-of-spec procedure must be in place and followed. We need to have comprehensive investigations and CAPA follow-up. All out-of-spec results must be documented. The documentation must be to an appropriate standard must be easy to follow. There must be a logical flow. We need to see the use of comprehensive checklists and protocols used for hypothesis, and any additional testing must be justified. You have to justify everything. Out-of-spec results and investigations must be reviewed at regular intervals. Are identified issues, are they isolated or are they recurring? Are there trends? And are issues investigated and closed in a timely manner? Just looking at some of the resources that were used in preparing this presentation and that can be used in generating procedures. In the European Union, obviously it's UGLEX volume 4. The United States, the FDA guidance in 2006, investigation of out-of-specification test results for pharmaceutical production. United States versus Bar Laboratories, it's always worth a look at the Bar case. MHRA has some excellent guidance on out-of-spec investigations, and people who are familiar with the MHRA guidance will recognize that I made use of some of their flow diagrams in my presentation. And there's also a WHO guidance on testing of suspected and falsified medicines. So I'd just like to thank you for your attention and wish you all the best in your future investigation of out-of-spec results. So thank you very much.